Hello students. Today we will discuss about the genetic recombination in bacteria. Sexual reproduction has long been thought to be absent in bacteria. Recent experiments with the help of electron microscope, however, have demonstrated the exchange of genetic material followed by genetic recombination in some species of bacteria. This is the essential feature of a sexual process. Bacteria are able to exchange genetic material by exotic means as well as by a straightforward conjugation process which closely resembles sexual reproduction in other organisms. There are two principal methods of bringing about genetic changes by exotic means in bacteria. In both these, there is transfer of DNA from one organism to another without the two ever coming into contact, but the technique employed is dissimilar. And these two phenomena are transformation and transduction. First of all, we will take transformation. The first transformation experiment was carried out with pneumococcus, which causes pneumonia. The bacterium is oval in shape and is surrounded by a capsule. Occasionally, a pneumococcus cell loses its capacity to secrete a capsule around it. This inability is transmitted to all its progeny. In this way, a clone of non-capsulated bacterial cells is produced. These clones of pneumococci have lost their virulence and do not cause disease. On a solid medium, these non-capsulated, non-pathogenic pneumococci form colonies which appear rough. The capsulated or encapsulated pneumococci, on the other hand, form colonies which appear smooth. It has become customary with the bacteriologist to apply terms rough and smooth to the pneumococci without or with capsules. The former are denoted by type 2 and the latter by type 3 pneumococci. Now, a British bacteriologist, Griffith, in 1928, injected mice subcutaneously with a mixture of type 2 rough non-pathogenic and heat-killed type 3 smooth pathogenic bacteria. Neither of these would have been harmful to the mice. On the contrary, the mixture killed many animals. The animals that died were found to be infected with the capsulated smooth pneumococci of type 3. The result was astonishing because type 3 organisms which were injected had been killed by heat. It was obvious from this experiment that association with the dead smooth type 3 bacteria had in some mysterious way restored virulence of living rough type 2 bacteria. On examination, the bacteria from the dead mice were found to possess capsules and formed smooth colonies. It is evident that the association transformed living type 2 bacteria into type 3 bacteria. The fact that the progeny of the latter were all type 3 showed that it was not due to their having surrounded themselves with capsules of the dead ones. In all similar experiments, the acquired property was that of the dead organism. In other words, the dead smooth bacteria transformed the living rough bacteria into their own capsular type. The substance in dead capsulated bacteria responsible for the transformation has subsequently been isolated and is found to be deoxyribonucleic acid DNA. It has also been found that transformation is not dependent on the presence of dead bacteria. It could equally take place with cell-free extracts from smooth bacteria. From the account given above, it is clear that the transformation may be defined as the transmission of heritable property from one cell to another by way of free naked DNA without the recipient and donor cells ever coming into contact. Next method is transduction. It is widespread phenomenon which differs from transformation. It is the mode of genetic transfer. In transduction, genetic transfer is carried out by the agency of a bacterial virus known as the bacteriophage. Only temperate as opposed to virulent bacteriophage can act as agents of this transduction. Transduction, this is a genetic transfer mediated by bacteriophage. The phenomenon of transduction has been studied in Escherichia coli with the phage vector P1 and Salmonella typhimurium with their vector. The bacterial cell infected with a temperate phage is termed as lysogenic. Here you can see in the diagram that the bacteriophage has infected bacterial cell. In A diagram you will see. Then it is the coliphage. The first one is the infection phase, then the latent period phase. In this, the virus DNA, and they've all disappeared. And then in the second phase, you will see that the synthesis of the components of the virus, protein code is formed, and then is the formation of this. That is a genetic recombination between the bacterial DNA and the viral DNA. And after recombination, again there is the assembly of the phage components, and then is the liberation phase. So by this, by infection of the bacteriophage, there is the recombination of the recombination of two DNAs, and this process, it is called as the transduction, and this we have already discussed under the multiplication of virus. Then is the conjugation, bacterial conjugation. It has been studied in colon bacillus, Escherichia coli में इसको पढ़ा गया है. This is the E. coli. There are two mating types of cells in E. coli. Both are haploid. One of these is a donor or the fertile cell. You can see the donor cell, and other is the recipient or the receptor cell. The cells of the opposite types come together in pairs. The donor cell has a special kind of pili known as the sex pili, which help to attach it to the cell wall of the recipient cell. A conjugation bridge or tube develops between the two conjugates at the point of contact. It appears that during the conjugation process, the ring-shaped DNA strands breaks at a definite point and becomes a filament. Without this, the transfer of genetic material cannot take place. However, it still remains double-stranded. The genes A, B, C, D, E, F, G, H, I, here you can see in this diagram, are arranged on it in the linear series. It was first shown that the gene A of the donor cell moves into the recipient cell at about 7 minutes. This is followed by B at 9 minutes. And consequently, all the genes, they are transferred within 18 to 25 minutes. The conjugates stay jointed for a short time so that only a portion of the DNA strand of the donor cell could get transferred to the recipient cell through the conjugation bridge. The recipient or ex-conjugant or zygote is thus partly or incompletely diploid. As you can see in this diagram, it possesses a complete recipient chromosome DNA, but only a part of the donor chromosome. You can see in this diagram, D diagram. In the recipient, and there is a entire DNA and a part of the donor DNA. When the progeny of zygote are grown, it is found that the cell is recombinant. It grows under the conditions that will support neither of the parents. This partly diploid state of maintained only for a short period. The zygote cell recombinant shortly discards the fragment of the donor chromosome and reverts to the haploid state. Thus, bacterial conjugation differs from true sexual reproduction in two respects: that absence of meiosis and formation of a partly diploid zygote. After transfer of genetic material, the donor cell must perish. Now, scientists suggested that only one strand of the donor's DNA enters the recipient cell, as it does synthesis of complementary strands takes place, At the same time, a new DNA strand is synthesized on the donor DNA to replace the strand which has been removed. So, this was all about the genetic recombination in the bacterial cell, which includes transformation, transduction, and bacterial conjugation.